Perfect. So good morning, everyone. I'm going to talk about my PhD project in general, about microplastic pollution, and specifically about this part of the PhD project, the microplastic ingestion by Zebrafish, Danio, rerio. Sorry, I'm just going to lower this one here. Okay. So as we all know, plastic pollution is a big problem nowadays, and with the amount of coverage that has been given from social media, we need to be very careful and pay attention to it. So we have microplastic items, the big ones, and then we can have microplastic items that can be primary or secondary microplastics items. The primary ones are produced to be of a microscopic size scale, and the secondary microplastics are actually derived from degradation and fragmentation of the bigger ones. The main polymers produced nowadays are polypropylene, polyethylene terephthalate, and polystyrene. And after the huge amount of production, some of them can be incorrectly disposed of and end up in the environment. And traveling through the rivers and estuarine system, they can get from the mainland to the beaches where the fragmentation and breakdown into smaller plastic fragments can happen and then enter the environment and ocean. We know that they can have a number of negative effects on all animals and biotas into the ocean and environment. Some of them are absorption of pollutants. So these plastic items can actually absorb pollutants, persistent organic pollutants, while they're traveling into the mainland to release them into the stomach of the animals once the animals ingest them. So that can lead to an intoxication. They can also be a vector for alien species. So we have alien species that can hop into plastic debris and then get transported into new environments and be, again, an invasive alien species and and can lead to a biodiversity loss. They can also be ingested by a number of animals, and through the food chain, they can biomagnificate. So through the food chain, they can add the number up and become even a greater problem. We can also have entanglement and drowning by marine mammals and marine turtles that can usually get entangled into these ghost nets. And we can also have effects at all levels of the food chain and trophic chain. So starting from the phytoplankton, we can have oxidative stress and a lower photosynthesis production. Then we can have into the zooplankton level an ingestion. And also on a higher level, we can have zooplanktiferous fishes that can mistake the microplastic for the zooplankton because they're quite similar in size and shape and ingest them instead of the real food. It's a problem that is a global threat nowadays. It has been found that there are mainly 5 plastic islands, very big, and they are corresponding into the 5 plastic gyres into the oceans. So it has been found also in the North Pole. So we can see that the plastic is basically going everywhere through the pathways of the currents into the oceans. So that's why it's a global threat. This part of my PhD, unfortunately, I'm just on the pilot studies because of coronavirus and disruptions, but I'm studying the microplastic ingestion by Zebrafish, Danio, rerio, and I'm using this model organism because it is the vertebrate model organism and an emerging one for microplastic studies. The aim of the first pilot experiment is to investigate under laboratory conditions the ingestion of plastic by fish to determine whether certain microplastic characteristics like size and density can impact on incidence of ingestion, accumulation, and fish death rate. So I've done that with the first part of the pilot using 3 tanks, one with PP, polypropylene, the other one with polyethylene terephthalate, and the third one was the control one. We have found quite a lot of microplastic that was ingested. So here we can see the picture from the polypropylene after 24 hours, polyethylene after 168 hours, the end of the experiment and last sampling day, and polypropylene at 168 hours. These are all the pictures taken from the stomach dissections. We can also see in here a picture of actual plastic fragments inside the stomach and then after the dissection and extraction. We can also see the plastic fragments being stuck in the gills, the second tissue that I've analyzed of the fishes. So I've extracted stomach content and gills, and we can see here how the plastic fragments, PET especially in PP, can be just stuck into the gills after the operculum was removed. The first data I had are evidencing are showing an actual ingestion of this microplastic by the fish. So about this graph, we're talking about a percentage of microplastic contaminated fish. And we can see that apart from this probably outlier at day 2, we have microplastic ingestion increasingly higher throughout the experiment and towards the end of it, with especially PET with the highest concentration in stomach tissues and PP with the highest concentration in gills tissue. And this can be seen also from this graph, that is number of microplastic fragments in contaminated zebrafish. So this has been analyzed about for the 3 sampling days where microplastic fragments have been actually found. And we can see here too an increase of microplastic pollution, so maybe and possibly an accumulation. And again, we can see that the PET, polyethylene terephthalate, is mostly present in the stomach tissue, and the PP, polypropylene, is mostly present in gills. So we analyzed also the microplastic size. The microplastic size that we fed to the fishes was of a 500 to 600 micron size range. And inside of this size range, we could observe a tendency to get a smaller plastic size range towards the end of the experiment. So another thing we would like to verify is that if they already ate all the big ones and were left just with the small ones. So this is very briefly what I've been doing. There is a second part of this pilot, but unfortunately, because of the coronavirus disruption, I wasn't able to analyze that data. I just got back to the lab, so hopefully in a couple of weeks I'll have that data too. And these are the next steps. So this is a part of my PhD, and we would like to run these set of experiments throughout this year. So we're going to do a proper set of experiments replicating these conditions. So we're going to analyze polypropylene and polyethylene in 2 different size ranges, 500 to 600 microns, and then a smaller one, 250 to 300 micron size range. And then I would like to look into the shapes. So if the shape of the microplastic is actually affecting microplastic ingestion, and I'm going to do a study with fibers versus particles. And then I'm going to analyze the colored microplastic, because we have quite some papers that are quite contradictory. The main theory here is that fish should be eating plastic that is colored in a similar way as their natural prey. So we're going to test that hypothesis. And last but not least, we are thinking about residence time or weather microplastic experiment. So the residence time would be an ingestion of microplastic to be then taken off the microplastic polluted water, and to analyze if the microplastic is going to be stuck into the stomach and gills, or if they can just get rid of that. And the weather microplastic will be an experiment about we feed to these fishes weather microplastic, because apparently in the wild has been found that they should prefer weather microplastic because it releases some sort of chemicals that it can attract the fishes. So these are the next steps. And the other branch of my PhD project will be about tumbling, but that's another thing. And we're going to talk about it another time. Thank you very much for your attention.